Gene interactions can change the expected outcome of crosses. In a 1945 trial, Joan Barry accused Charlie Chaplin of fathering her child. The jury declared that Chaplin was the child's father, but the newspapers commented, "California has in effect decided that black is white, 2 and 2 are 5, and up is down." The disbelief that Chaplin fathered the baby was based on hereditary laws. The mother was blood type A and the baby blood type B, which meant that the baby's father must have type B or AB blood, and Chaplin, however, had type O. But the jury was right because of epistasis. Epistasis is a gene interaction where a gene's allele masks the phenotypic effect of another gene. The gene that masks the expression of the other gene is the epistatic gene. The gene it masks is the hypostatic gene. The H gene interacts with the gene for ABO blood. In the Bombay phenotype, the homozygous recessive H allele masks the effect of allele A and allele B of the ABO blood type gene. Because the H antigen is the precursor of the ABO blood group antigens, people with homozygous recessive H will not produce the ABO blood group antigens. Thus, a blood test for ABO blood type would indicate people with the homozygous recessive H genotype as having blood type O, although they could have the alleles to produce antigens A and or B. So in the case of Barry versus Chaplin, it would be possible for Chaplin to be the father. If Chaplin were homozygous for the recessive H allele, a blood test for ABO blood group would show Chaplin to be O, although he has the B allele. And baby Carol Ann, inheriting the B allele from her father, would express the B antigens as she inherited the dominant H allele from her mother. By the way, if people homozygous recessive for H need a blood transfusion, they can receive blood only from the dominant H deficient donors. A transfusion of normal group O blood can trigger a severe transfusion reaction, as O-type blood has antibodies that react with the H antigen. Another unexpected inheritance pattern was seen in the case of two albino parents having normally pigmented children. Albinism, or a lack of skin pigmentation, was thought to be caused solely by one recessive mutation in the tyrosinase gene. So people wondered what the explanation for this case of albino parents having normally pigmented children could be if the parents were the biological parents. It was found that more than one gene was involved in skin pigmentation. Each albino parent was homozygous recessive for a different gene involved in skin pigmentation, so the children received a dominant allele of each gene from the respective parent and could express pigmentation. Where an allele for normal function is required from two different genes to express the trait, the genes are said to complement each other. The interaction is called duplicate recessive epistasis or complementary gene action. Gene interaction does not negate Mendel's laws, but they change Mendel's dihybrid ratio. In standard Mendelian genetics, where two separate genes are inherited independently, the expected outcome of a completely heterozygous cross involving two non-interacting genes is the classic 9:3:3:1 ratio. To see how epistasis changes the Mendelian ratio, predict the phenotypic proportions expected in the progeny when dihybrid white dogs are mated together. But first, some background on how genes involved in coat color function. The big I allele, which causes inhibition of coat color pigment, is dominant and prevents the expression of color alleles at another independently assorting locus, producing white coat color. When the recessive homozygous condition exists at the inhibitor locus, the alleles of the other locus may be expressed. The presence of a dominant B allele produces black fur, and the homozygous recessive B condition produces brown fur. Now that you know how the genes influencing coat color function, Big I suppresses color, small I, color can be produced, big B, black, small b, brown. You should be able to select the phenotypic proportions expected in the progeny when dihybrid white dogs are mated together. Is it A. All white, B. 9 white, 6 black and 1 brown, C. 9 white, 7 black, D. 12 white, 3 black and 1 brown? As observed in the Punnett square, there will be 12 white, 3 black and 1 brown. Every puppy with the dominant allele for coat color suppression will have white fur. When the puppy is recessive homozygous for this gene, i, the other gene will be expressed. So a dog homozygous for I and having the dominant allele for black fur will express black fur. Only dogs homozygous recessive for both genes will express brown fur. Unlike in the example of the Bombay phenotype, in this example, the gene that masks the effect of the other is dominant, and only one copy is required to mask the effect of the other gene. Thus, it is called dominant epistasis. The Bombay phenotype is recessive epistasis, since a homozygous recessive genotype masks the alleles at a second locus. When there is recessive epistasis, a dihybrid cross results in an offspring phenotypic ratio of 9:3:4. Often, the biochemical basis of epistasis is a gene pathway in which the expression of one gene depends on the function of a gene that precedes or follows it in the pathway. To see how genes in the same biochemical pathway give rise to epistasis, determine the phenotypic ratio among the offspring of a dihybrid cross big A, big B, little b, crossed with big A, big B, little b, based on the following information. Dominant alleles A and B produce the functional enzymes A and B respectively. Enzyme A catalyzes the conversion of compound 1, which is white, to compound 2, which is red. Enzyme B catalyzes the conversion of compound 2 to compound 3, which is purple. Recessive alleles A and B do not produce enzymes. In this reaction, one copy of the dominant allele produces sufficient enzyme to catalyze the reaction. The answer to this question is a phenotypic ratio of 9 purple, 3 red, and 4 white. The explanation is as follows. If the plant is homozygous for the recessive allele of gene A, enzyme A will not be produced to catalyze the conversion of compound 1 to compound 2. And since enzymes work on specific substrates, enzyme B, even if present, has no substrate to convert to compound 3. Hence, all plants homozygous recessive for A will only have compound 1, and the flowers will be white. If the dominant A allele is present, enzyme A will be produced and convert compound 1 to the red compound 2. If the plant has the dominant A allele but is homozygous for the recessive B allele, the absence of enzyme B will prevent the conversion of compound 2 to compound 3, and the plants will have red flowers. Only when gene A and gene B's dominant alleles are present will both reaction steps be catalyzed to produce the purple compound 3. In summary, epistasis is the gene interaction in which alleles at one locus mask the effect of alleles at a second locus. The gene that masks the expression of the other gene is the epistatic gene. The gene it subordinates is the hypostatic gene. Epistasis may be recessive, dominant, double recessive, or each type of epistasis results in different phenotypic ratios, such as 9:3:4, 12:3:1, and 9:7. Now let's check our understanding of epistasis with a few questions. You may pause the video while you think about your answer. Question 1. How many locus or loci are involved in epistasis and dominance respectively? A. 1 for epistasis and 1 for dominance. B. 1 for epistasis and 2 for dominance. C, 2 for epistasis and 1 for dominance, D, 2 for epistasis and 2 for dominance. The answer is C. There are 2 loci for epistasis since one gene's allele masks the alleles of another gene. And there is 1 locus for dominance since dominance refers to an allele of one gene masking the effect of the other allele of the same gene. Question 2. In poultry, the shape of the comb varies greatly and involves at least 2 pairs of alleles. The allele dominant R can result in the rose-shaped comb, and the allele dominant P can result in the P-shaped comb. If both dominant alleles are present together, gene interaction produces a walnut-shaped comb. Single comb types can result when a bird carries both recessive alleles in the homozygous condition. Can this interaction be called epistasis? If not, state why. The answer is that this example does not show an epistatic interaction because you can distinguish which alleles are at each locus. For example, comb shape of poultry homozygous recessive for R but having dominant P is different from comb shape of poultry homozygous recessive for both R and P. However, this is still a gene interaction. Question 3. Deafness in human beings is inherited as a recessive condition. 2 deaf individuals have married and produced children with normal hearing. Propose a biological explanation. The answer is that the parents, individuals 1 and 2, must be homozygous for recessive mutations in different genes. That is, one is little a, big B, and the other is big A, little b. None of their children is deaf because all of them are heterozygous for both genes. That is, big A, big B, little b. Refer to the videos on genes and alleles as well as dominance to refresh your ideas on these related concepts. Thank you for listening.